Hey Andy, thanks for coming out and joining us on the Baobab project, where we explore the wild world of wellness through the lens of the Africa's iconic superfruit baobab. So, for everybody that's listening, this is Andrew Benson or Andy Benson. You're the head of the Food for Health Center.
Yep.
We'll start with that because I think that's the big passion for me. That's how I actually met you. You're also a professor of biotechnology, we'll call it.
You can call it that. That's
Whatever.
fine.
Professor in the Department of Food Science. I think you do everything, so I don't really want to pigeonhole you because from every conversation that we've had, I feel like you do every part of food science.
Well, I'm not even trained as a food scientist. I'm trained as a microbiologist. That's my original training. And I ended up, long story, in a food science department and spent several years studying food safety. Microbes that kill you, right? And looking at genetics and evolutionary processes that were driving those organisms and how they were ending up on our food and getting into us, and what did they have genetically that allowed them to do that. In about 2006, 2007, next generation DNA sequencing technology was commercialized, and that's all you need to know about that, right? Because we and me and a couple of other folks in the food science department recognized, and we weren't the only ones, people around the world did, recognized that technology would become an analytical tool that would let us enumerate microbes in very complex communities. Like for 200 years, microbiologists have known you have these complex microbial communities in the gut and the water and all over the place. There's no good way to study them with the traditional methods.
Is it the idea that there's just too many at once?
There's too many at once, and with the old culture-based methods where you're culturing a single species at a time, there was just no good way to do it. It was too hard to do. You couldn't begin to, if you think of a rainforest as a great example, you couldn't count the trees or figure out which species were there. There was just no way to do it with a microbial community. DNA sequencing let us do that. The next generation DNA sequencing allowed us to do that. And I don't want to bore you with all the details, but it blew the door open to the field because it became the analytical tool that let us go in and start understanding how many organisms, which organisms are there, what's the variation look like across a population of people. Is yours the same as mine? Or all of that was low-hanging fruit in 2006, 2007.
And so, generally speaking, we have a good idea, or we know.
We know a lot more now, but back then you couldn't name 100 species that were in the gut. Nobody knew back then. We just started learning which organisms are actually there because that technology allowed us to do that. And being in the context of a food science department, one of my colleagues and I started thinking about, we've got this analytical platform that's available now. Can we use that in a way to really understand the gut microbiome? And that word was coined around.
What was it
that
before
time as
that?
well.
Like what was the, because I talked to Jacques about, when we're going to talk about this more, but
Yep.
gut health and microbiome, right? And they're thrown around now. So I think people know what the microbiome kind of generally means now. Gut health is starting to make sense to people in a general sense. But what was it actually called or even talked about before?
Generally we'd call it the gut flora or microflora. Was kind of the word that we used.
It's good pivot in branding.
Yeah. It was not a great word, but that was kind of, and it was this vague, sort of term that you use, knowing that there's a complex community there. We have no idea how complex it is. We have no idea what it really looks like. There's only a few species that had been cultured and studied very well. So that platform, that analytical platform of DNA sequencing really blew the door open to the field.
Well, it sounds like it took you. So you were studying the things in your gut that were going to cause you to be sick.
Yeah, things that get on food that get in your gut.
Yes.
Yes.
And then with that sequencing, you continued to do that for a while longer?
I did it for a little while. I think by 2009, and I still do a little bit of it, but I shifted my whole research into the gut microbiome at that point in time because, we figured that if we could really understand that gut microbiome and the ecosystem that it's in, and we're in a food science department, we could actually think about designing foods that feed not only your body but also the microbiome. right? And that was the whole thing that drove us. And so we started off 2007-ish. We became known as the Gut Function Initiative. For many years, we were known as that. That's when we hired Jens. Walter was one of our first hires. We hired Dan Peterson, who was a mucosal immunologist. Dan set up our original germ-free mouse facility at the university. And we ended up buying one of these next-generation DNA sequencers for $500,000. Now, paperweight, but it's amazing how fast that technology has advanced. But very early in the game, we were an entity. And so we spent those first, I look back on them as the golden years, because everything was really low-hanging fruit. You know, I can remember looking at the first set of microbiome data that we generated and going, oh my God, I have to pinch myself. Am I really looking at this? This is so cool. You know, people dream of having something like this.
Like no one had ever seen anything like it before.
Well, no, I mean,
we
as a scientific community were just starting to see it for the first time, right? And so then we were starting to learn, okay, how do we deal with this data now? Because it was massive amounts of data. And so we had to learn how to do bioinformatics and learn how to use computation.
What is, explain that a little bit. I know it's probably a little bit in the weeds, but what does that mean? I mean, it's pretty much computer science at this point, correct?
Yeah, it basically is. Science. I mean, it's data science, because you're generating this next generation DNA sequencing technology. And again, I don't want to get too far in the weeds with that, but it generates massive amounts of data, right? Pretty large scale data, at least for biology it is. And so you need computer science and computer science-based tools to actually process and analyze this data. Prior to that, the amount of genome data we had was small compared to what those next-gen DNA sequencers would generate. And that was why it was such a game changer, is because it became much less expensive and you could generate these massive data sets that would allow you to identify and enumerate I would say all the organisms, but you know, the most dominant organisms in a microbiome.
Okay, so I guess to start off with, you start to identify them, but identifying them is just the first part. Well, I guess you knew what was growing on something or what was on a food item that could get somebody sick,
right.
And it's going in the gut. And then we generally had a couple ideas of what's in the gut, but not very much. And then so now you start to know what these bacteria are. Are they connected to outside the gut bacteria as well? So then that way we know potentially how they function. Or
at some level, but I kind of put the food safety stuff aside at that point and went whole into what are the factors so what's the microbiome look like? What does it look like across populations of people, right? How much inter-individual variation is there? And it turns out there's tremendous. It's just yours is unique as your fingerprint is. And then what are the factors that shape that? You know, what makes your microbiome? How does that come to be? And we're still studying that. But my initial focus was on how does your individual genetics influence it? That was one of the things I studied.
Yeah, Generally, this is how you are, but there's just so many external factors.
Correct. And
did it go pretty quickly from, all right, this is genetic to, oh man, this is everything else?
You're almost spot on, right? So I spent a good bit of time studying the impact of host genetics early on, and the answer to your question there is that you can measure it. You use quantitative genetic approaches where you're screening large numbers of individuals and using statistics to identify it. But the problem was that all of the other factors, especially dietary factors, would swamp out your genetic signal. You could completely swamp out genetic signal with a diet
pretty
change.
quickly.
Fairly quickly, yeah.
I mean, I think you see this, right? When we think about talking about diet, right? Western diet versus native diets versus, like forager diets, like how quickly people come to a different country. I mean, America's a perfect example, but like people that come from another country.
If you look at immigrants, yes, and you look at first generation and then look at the second generation, it's amazing how fast that change happens.
It's like not even the same person. Not even the same.
The microbiome will change over time and adapt as the dietary pattern changes.
Huh. Okay. I'm starting to get this. I think of, we've kind of been talking from the mouth to the, as Jacques or David would joke, the anus, right?
Yeah,
This
yeah.
is like
It's a
where we're
tube.
thinking,
It's a
it's a
big
tube,
tube.
right? Explain your thoughts on the entire idea of the microbiome, because I think you have a really good, simple explanation for people, because I think people are starting to hear it, right? They're seeing it on packaging, they're knowing that they should be doing something to improve theirs, but a lot of times then they're sold a product that just says, hey, this does this, right?
Right.
And like I've told everybody else, like, we're doing this not because, hey, drink Tiiga, buy Baobab, buy Tiiga. It's like, I genuinely want to understand this, but also I think they need information from people that don't have time to start companies. Well, they do have time to start multiple companies, but their companies are focused on research and finding these things out. So let's start with just the idea of how you kind of present the microbiome as
Yeah.
well. I do want to talk about.
From the mouth to the anus, you actually have multiple microbiomes, right? So you have an oral microbiome, right? It's its own ecosystem, and and it's not just the microbes that are there. Remember, the microbes are in contact with your tissues, right? And so there's interactions that are going on, very complex interactions that are going on. So it starts in the oral cavity, and by the time you get down to the stomach, then the numbers fall because the stomach's very acidic. So there's very few organisms that are actually in the stomach itself. As you move into the small intestine, now the numbers start to grow again. And so in the small intestine, you've got around, I would say, millions to billions as you move from proximal, meaning close to the stomach, to distal as you move close to the colon. You go from millions to literally billions of organisms. And then by the time you get into the colon, it's trillions.
So do they? Okay, so you have a lot in the mouth throat area, and then as it goes in the stomach, do they die off?
Oh, yeah.
But then enough survive.
Well,
But is
that's
it like a
big question.
bottleneck? Like, that's a
Yeah.
really interesting idea. Like, do
Yeah.
they bottleneck, and then only specific ones that can make it through the acidity process?
It's a really good question. What we think is that obviously they must, right? And if you think about it, they get buffered too, right? So if
Yeah.
you're eating food, that'll buffer that. Right, that the pathogens take advantage of that too. So that buffers that stomach acid, and they can get through that way. But then the key is that when you're born sterile, right? You're in the uterus, you're sterile.
Because no oxygen,
There's
correct? Or is that? Or
well,
am I
it's not
just-
so much that there's no microbes.
Mic-
There's no microbes
zero
in,
micro-
in the uterus. That is a sterile. The womb is a sterile environment, right? And so the minute you're born, as you're going down the birth canal, you're starting. The organisms start coming in from mama, right? And so when you're born, you have a fairly simple microbiome, a simple microbial community, and then depending on whether you're breastfed or bottle fed, that makes a sort of a crossroads as to how your microbiome might develop. If you're breastfed, there are things in breast milk called human milk oligosaccharides, or HMOs. That doesn't stand for Health Maintenance Organization for our discussion. This is human milk oligosaccharides, and those HMOs, the baby's
Sorry,
body.
It sounds better.
You get tired of saying it.
Yeah,
You say HMOs, right? But those HMOs, the baby's body cannot utilize on its own at all. And here, mama's pumping all of her energy into making HMOs to put into the breast milk, right? Why is that? Well, there are a very small number of gut microbes that can degrade those HMOs. And one species that's well known for that is called Bifidobacterium. Bifidobacterium longum, subspecies infantis, is one of them that can do it. Small number of these organisms that can do it. And so early in life, if you're breastfed, those organisms dominate that microbiome. And because they're thriving on that breast milk and the HMOs that are in that breast milk, and they can selectively grow on it, and it isn't until the child starts weaning that microbiome starts to become more complex, because those HMOs and the organisms that can degrade it just dominate the microbiome. As the baby starts to wean, now other organisms are able to start taking up that niche space. This Bifidobacterium species that I mentioned, it's great at growing on HMOs, and it can grow on some plant-based carbohydrates, but not as well as other species can. So you start getting these other species moving in that will displace those original species. This is called a succession in ecological terms, where you have a wave of new organisms coming in and they start to colonize, and then depending on how that baby.
Do they destroy the other ones?
I don't know if destroy is the right word.
They just move into the new feed.
They just
outcompete.
outcompete them.
Okay.
Yeah, the other ones, they can't grow as well on the nutrients. The ones that are genetically geared to do it, they grow well on it and they take over the ecosystem. So by the time you're into adolescence, you've now developed what's going to be your microbiome, right? And that microbiome is fairly stable. I mean, it's remarkable how stable they are.
Starting at like what age?
Oh, good question. I'm going to just say it's
roughly.
It's in adolescence, right?
Yeah,
it's in
that's
adolescence,
crazy.
right?
Really?
Yeah, because during development you've got other things going on, hormones and stuff going on that can impact that microbiome. But then once you reach adolescence, it starts to become stable, and once you're an adult, that microbiome is essentially stable as a brick. Now, that doesn't
That's
mean
your genetic microbiome at that point. Or is this still...
it's not your genetic. It's assembled itself, right? It has assembled itself through ecological processes. It's assembled itself in your gut at that point.
Okay.
And yeah, your genetics can influence it a little bit, but a lot of it's influenced by your diet and exposure to antibiotics. How much previous exposure you had to antibiotics, because those ding the microbiome as well. They don't just kill the bad guys. Antibiotics also will impact some of the good guys.
I think everybody feels that when they when you can't go to the bathroom for like a week after taking the antibiotic.
Yeah. It's so that early life exposure to antibiotics can really impact that process, and even later in life it can impact it. But by the time you're an early adult through adolescence, that microbiome is very stable. Like I said, there's daily fluctuations and perturbations, but for the most part, it's stable. And you know, you can kind of think of it as like the blob. You know, you can poke it, it'll change shape, but it'll come back to its original shape, and it sort of maintains that ecological dynamic and that function. And what's important about that is, you know, those species have now evolved and adapted to living inside that gut ecosystem. The whole thing is an ecosystem. And those species are interacting very intimately with your small intestine, with your large intestine. And when you refer to gut health, right, it's not just food going through and being digested and pooping out. No, there's a lot more going on than that when you speak of gut health. You know, gut health really refers to the function of the gut from the mouth to the anus, which is different. There's very different functions in the oral cavity versus down in the colon. And one of the major things that the gut health really speaks to that most people don't think of is the interaction between this dense population of organisms that are in the colon and the immune system. So 70% of your immune cells are in the gut. So a big portion of your immune system is hanging out in the gut. And there is a very thin layer of tissue that separates the immune system and this dense population of microbes.
What is that thin layer?
It's called the colonic epithelium, and in the colon, or in the small intestine, same thing. But you've got this very thin layer of cells that separates these two. And it's almost like having two armies on either side of a fence. Like if you break that fence down, all hell's going to break loose.
And how does that fence break down?
So
does it break down? I guess
it can. Yes. If you heard a leaky gut, that's what that is.
That's what I was about to ask. It sounds like what people have been throwing around as leaky gut.
Yeah, we call it barrier integrity, right? Maintaining the integrity of that barrier. So that barrier needs to be able to absorb nutrients, which there are very specific receptors
from the immune
cells.
side.
Well, from the inside.
From
right?
the inside,
where the food is on the inside, the food's going through with the microbes, right?
Okay.
And microbes are also helping to degrade the food, especially down in the colon. The body is absorbing things. It's digesting some of it on its own. It's absorbing that. It's also absorbing things that the microbes are digesting and secreting, the metabolites the microbes are making. So that interaction needs to happen, right? It needs to be efficient, our bodies have evolved to be very efficient at pulling those nutrients out of that ecosystem. And to do that efficiently, the cells need to be a little leaky, right? Just a little, right?
It's like the permeability needs to be there for a reason, but
there's
not,
a reason.
It's not a waterfall.
There's a reason, exactly. But what can happen is when the microbiome becomes messed up, and you've heard this word dysbiotic or dysbiosis, right? That's a microbiome whose species configuration is goofy, right? It's not.
What does that mean though by goofy, in the sense that it's got like processing specific ones, and then
it's got too many of the wrong organisms.
Yeah. Okay.
Yeah. So it's got too many of the wrong organisms, and those organisms tend to cause that barrier integrity to break down a little bit, and then bits and pieces of those organisms, as they die, get through there, and they inflame. They get the immune system all jazzed up and inflamed, and it's just this sort of vicious cycle.
I feel like that's where autoimmune stuff.
Well, there
that might
is
be a different completely subject. But.
Many of the what we call complex lifestyle diseases. Right.
That's a really long way to describe complex lifestyle diseases.
So these
I didn't know that was called that, but that makes sense.
So these are Western diseases,
that are solved with a pill now.
Well,
we'd
like to, but metabolic diseases such as diabetes, some cardiovascular heart disease, inflammatory bowel diseases. These are things you see that are predominantly in the West that you don't see in developing countries so much, and part of that, we believe, is due to this disconfiguration of the microbiome, and that's actually contributing to the disease process, and this inflammation that it begins there, right, in the colon, but it goes throughout the body, and so the metabolites that the microbes are making not only make their way to locally right on the other side of that colon, but they find their way into organs and other places, and so inflammation starts happening there. And you can see how that starts triggering and exacerbating some of these disease processes.
Can you explain how inflammation affects the body? This might not be your area, so this isn't a good question. But because I think it's still antioxidants and inflammation. I think for people, they're buzzwords, but they don't register how they affect the actual body or the organs. Excuse me, like organs or tissues.
Yeah, that's not really my.
No, okay. So I'm going to hold that. So I don't need to ask you this, but that makes me think about it, because that's another one that I want to find out, because I want to understand the inflammation part. But we know that now it's triggered in that process
it's
through
exacerbated.
Exacerbated, or it can be triggered through that, and now it's in the body, and now this inflammation's around. Okay,
right.
That makes sense. But then that also makes sense then. We keep on trying to solve a lot of these, the complex, I guess, what was it?
Complex lifestyle diseases.
Complex lifestyle diseases. So you have these complex lifestyle diseases, and it's like we continue to try to fight them, instead of being proactive, we're reactive, right? One. But two, we continue to try to fight them with something that's not focused around dietary changes, which is fascinating to me, especially when I continue to talk to people that study the microbiome, or just the literal, the microbiome track. Like this entire system is fascinating. So a couple questions I have. First one would be, actually, let's go, you said that it's very different between, and the only reason I say this is we have a 4-month-old, and so the difference between breast milk and then formula.
Right.
What happens in the formula process? Because now they're starting to get. It sounds like they're going to get bacteria earlier.
Well,
or different bacteria.
They get different, right? So their microbiome will look a little bit different. They'll still go through this succession. They'll have a microbiome that looks different than a breastfed infant, and the organisms that will dominate are organisms that can break down what's in the infant milk. And what you're finding now is that the infant formula producers are now starting to integrate these HMOs into the infant formula, which is a smart thing to do, right? So you can still, if you get the right ones that have these HMOs in it, then you can still get the benefit of having those HMOs in there and enriching for those organisms. But if you're buying a brand that doesn't have that, then you won't have that. But their microbiome will still develop along a similar path. It's not the same, but along a similar path. And then as you start to wean, then it'll get more complex again. And so by the time, and we don't we don't have enough data to really
Yeah.
understand this, but by the time those, let's say you had two groups of infants, one bottle fed and one breastfed, by the time they're adults, they look more alike than they did when they were infants, let's put it that way. So there's still an assembly process of this microbiome that's happening. And you still go through this succession it's because it's driven by diet. The diet's driving most of that.
Okay, so then that leads me to a question into the idea of these aren't complex lifestyle diseases, but behavioral things for adolescent and children.
Yeah.
And you obviously don't study the behavioral side, but it's interesting because now inflammation is going to change, and we were going to talk a little bit about this, but just like your disposition in general, and then the gut health to your mental health, or the gut-brain connection versus this leaky gut barrier. So you have the leaky gut barrier, and then you have a gut-brain connection, and it sounds like this is developing to get to a certain microbiome by the time you're an adolescent. And in that process, it's going through ups, downs, hormonals, and all those other things. But with a diet that's not helping this microbiome to reach this goal, or this future microbiome,
so
I think you got where I was going.
I know where you're going, right?
Because I'm super interested in this, because
still,
it's like we have so much stuff happening now that never happened before.
And our understanding in this area is still rudimentary.
Yeah.
You have to first understand that. But we know a lot more than we did, right? So clearly there are interactions between the brain and the gut. There's no doubt about that. Just think about the last time you got really nervous and all of a sudden had diarrhea, right?
It's pretty instantaneous. Well, I'm not saying
it can be, right?
Yeah, but it's crazy how that constipation, like
There's you know there's a gut-brain connection there,
and it always feels like it's the reverse way. It always never feels like it's you're producing something better for your gut than you feel. Like you don't think of it that way.
It's a two-way street.
Yeah, it's a two-way street, but you always notice it only from the things that you do mentally that change the way that you.
Yeah.
It's like travel, right? For me, travel generally constipates me more, and I don't know if it's because you don't think you can stay in the same routine, so then your brain's telling your body or your microbiome is shut down.
But do you get nervous about traveling? Do you have anxiety? I mean, I get a little.
I'm not a good person to ask that. I freaking love traveling.
Ask your wife, right?
Yeah, ask my wife. But but yeah, it's true. It's something. It's because you might not go to the bathroom at the same time, or you don't know if you what your day is going to look like. So yeah, I guess I mean, assume you just know that your day is going to be different. You know, you're not potentially go to the same locations. Yeah. You're not going to be in the same places. Interesting.
Okay. So there's that connection, right? And that's largely through a nervous connection, right? So there are nerves that run from the brain down into the gut. The vagus nerve is the primary one.
Yep.
And so there's an information highway that's going back there, and there's neurotransmitters that are flowing from the brain down to the gut. In the gut, there are also neurotransmitters that are being made by the microbes, right? So some of the gut microbes actually synthesize and produce metabolites that are identical to our neurotransmitters. So gamma-aminobutyric acid, or GABA, that's a good example of one. Serotonin, norepinephrine, all of those can be made by organisms in the gut.
Like they literally feed off of...
They're metabolites that they produce, right? So, it's fascinating. Now, most of those don't cross the blood-brain barrier, meaning that they won't get from the gut back up into the brain, but they can certainly act on nerves around the gut and then in the other parts of the body, right? So, there's this amazing connection where you have signals coming from the brain that can influence the gut and the gut function itself, and through that also impact actually the gut microbiome. So another case in point, remember I said we have this very thin layer of cells separating, yes, right? One of the things that's outside or inside the intestines, when the lumen, right, yep, inside the tube, the cells will synthesize a fairly thick layer of mucus, right?
Okay.
And the purpose of that mucus is to sort of help the digesta slide on through, right?
Yeah.
During the peristalsis, which is how the gut actually moves. It moves kind of like a worm does.
Yeah, like the accordion style.
Yeah, it's just very slow, right? That mucus helps sort of lubricate the lining so that the stuff will move through. But another function of that mucus is, again, to keep the microbes away from the actual contact with that thin layer of cells, right? To sort of keep a distance. make the fence a little bit wider, right? The signals from the brain can impact that mucus layer, and they go and they talk to certain cells, goblet cells in particular, that are in that lining that are secreting mucus, and they can either cause it to increase or decrease production of that mucus.
Like, give an example of like a thought, I guess. That doesn't make sense, but is it like literally a thought of being like, oh no, and that can change?
It's more like your anxiety, right? If you're having anxiety, that can impact that mucus layer.
How would you guys study this? This is incredible. You described it so well.
It's these neurotransmitters and these signals that come from the brain down into the gut area. They not only impact the microbiome, they're generally made to impact the cells, right? Your human cells that are lining the colon. They also affect peristalsis, right? So those the brain gut connection can impact that whole ecosystem, and including the microbes. But remember, I said it's a two-way street, right? We also know that certain things the microbes do, we don't even fully understand what it is, but certain things the microbes do can also impact how your brain's functioning. So great, you'll love this. So
this is what you knew I was going to ask this, because actually I was going to go, okay, let's go the other way. How does diet or what you're putting in your gut make this change?
You'll love this, right? So there are lines of mice, right? Breeding lines of mice that have high anxiety, right?
Wait, like this type of mice?
They have fairly high anxiety.
Yeah.
And then there's other lines that are just, they're couch potatoes, and they're low anxiety. They're just chill, right? So you can generate germ-free versions of those, and they start to lose those characteristics. Gets even better. You can take a microbiome from a high anxiety mouse and put it into a germ-free version of a low anxiety mouse, and that mouse will start having high anxiety just by transferring the microbiome itself. So clearly it's contributing to that process, right? And if the microbiome is contributing to that, then obviously the diet ultimately is one of the things that can be driving that. So that sort of walks you back too, right? And it comes back to trying to understand causality. That's something I wanted to make sure we hit today too.
Yeah.
With a lot of these associations, and people are hearing, the microbiome is associated with this disease and that disease. And what we don't know in many cases is the messed up microbiome causing the disease, or is it a consequence of the disease, right? Does the disease process, and in some cases it can be both, right? It's this vicious cycle, right?
Symbiotic.
But the reason we don't know for sure is it's difficult, because I can go study a healthy population and carefully control it as best I can, and study a disease population, and I can see that, there's a difference in their microbiome configuration. The disease population of people have this weird configuration of the microbiome. But from that data alone, I don't know what the cause or effect, which is the cause and which is the effect.
Yeah.
And so this is where these germ-free mouse models really come into play.
So explain that what happens with it. Now, there's some people probably that don't really get the idea, but at the end of the day, this is where you start most science and trying to understand. So explain how utilizing them, because you're able to create these germ-free test samples.
Yeah, so you can actually grow mice germ-free, right? And they'll breed. You can get them to breed, and once you get them into a germ-free environment, then they will breed, and you can have germ-free mice, right?
Wow.
And all their litter, as long as you keep it, they're in a
Germ-free.
bubble, right?
Yeah.
They're in mouse in a bubble, right? They're not normal, first of all. So you'll love this. They've got to eat roughly 30% more food just to maintain their body weight.
Because there's nothing.
Because there's no microbes to help break the food down. There's just whatever they can break down on their own, right? So they have to eat more food, which is.
It's weird, because I would have that's counterintuitive to me. I feel like they would have used less.
Mm-mm.
Because I feel like the microbiome, like the bacteria would like more food and break down better. But then I guess they're helping to help your body absorb better, correct?
So some of the products that the bacteria produce as a byproduct of them eating the food are actually used by your body.
Yeah, that's okay.
Yep. So
they can be more efficient, ironically, because
you're much more efficient.
you put in this little
harvesting.
bit, and now they're creating 10 different things that your body potentially needs
Yeah.
that didn't exist, that you can't do yourself.
Correct. So these germ-free mice aren't normal, right? Their immune systems are weird. Their behavior's a little bit off. You know, they don't develop normal. And if you give them a microbiome, you change all that, right? But
You can just like implant a microbiome.
Yeah, and so this is done quite frequently. So, case in point, if I want to understand if a messed up microbiome, a dysbiotic or disconfigured microbiome from the individual with a disease, if I want to understand if that is actually contributing to the disease process, you can take that microbiome and introduce it into one of those germ-free animals. And then you start asking the question, does that animal now start taking on characteristics of the disease?
Yes.
Right? It might not fully manifest, but does it start taking on characteristics? And in a lot of instances where those experiments have been done, that's what you see. You can actually transfer aspects of the disease just by transferring the microbiome. So you can take, one of the original ones that were done were from obese individuals. You can take an obese microbiome from an obese human, put it into a germ-free mouse, and that mouse will start taking on characteristics of obesity.
Is it like quick?
Fairly quick, yeah. I mean, like within weeks, yeah.
Okay, so
And you would obviously be comparing it to
a germ-free one or a normal
or
one.
germ-free, but also
Also
one
a healthy
that you colonize
microbiome.
with a healthy, right? And if you look at the weight gain, track the weight gain, and track other markers, you'll start to see that obese microbiome, the weight gains faster, and yeah, it's remarkable how that works. So these models have been very powerful in helping us to understand causality, and then also for studying mechanisms, because there we can actually start to begin to study the mechanism.
Explain that.
Well,
the mechanism of.
Well, so now, I mean, I can do experiments in mice that I can't do in humans.
Yeah, for sure.
Right. So, at the end of
Germ-free
what? Yeah. Well, not only that, but at the end of the experiment, the mouse can be dissected and whatnot, right? And so there's a lot of. I don't want to go into detail about that, but there's a lot we can do there. So we know that about this causality in some cases. So I told you the one about transferring anxiety. I mean, transfer obesity. So we know about that, but we don't know about that with all the diseases.
Okay, so you transfer an anxiety.
Mm-hmm.
You got an anti-anxiety mice over here. Can you transfer that microbiome back into the anxiety mice and make that like the chillest, most relaxed in between?
Yeah.
And obesity, same thing. You can have somebody that's...
Don't get me wrong, there's other factors, right? It's not just the microbiome. But the point is that you can transfer some of the characteristics with that microbiome. That's the thing to keep in mind. So it tells you that either properly configured or disconfigured microbiome is contributing to whatever process you're looking at there.
Can you then take a human microbiome in any way, shape, or form? Like, can we...
We put those into mice all the time.
So we're grabbing... So...
Yeah, it's called a humanized mouse model, right?
How do you get a human how do you get a microbiome? Like, how do you...
Stool samples.
Stool samples?
Okay. That's the most...
I figured that was it.
The most efficient way, yeah.
But, okay, so...
And it's a pretty good representation. Right?
Yeah, I mean, it's not going to be everything, because, again, we know that through that process, you're not going to have the same that would have been in the small intestine.
Right. Or
potentially even in the
It's mostly going to be the colonic.
Yeah, okay.
Mostly going to be the colonic.
So then, you obviously can't test this, but this is super interesting, because this sounds like the future, is then you take a human... good microbiome, we'll call it a good microbiome, or say like non-obese microbiome, and then you were to inject that into a obese person's microbiome.
Trying to change a microbiome is difficult. Let's go down this path.
Yeah,
so I want to talk about that, because so now you understand that these microbiomes can get messed up, right? And that they can help drive disease processes, right?
Yeah.
How do we prevent that, or how do we fix that, right? Can we do that? One of the downsides of these ecosystems is that in many cases, even the disconfigured microbiome is stable.
Well, that's what I was going to say. You're surviving, right?
Yeah,
like,
yeah.
Your body knows how. This is
Yeah.
what's so fascinating about your body. You can do anything, pretty much. I'm reading a book on people traveling through the Amazon jungle.
Yeah.
And all I can think about is, like, your body's going, what are we doing? But your body will, like in one part, this guy's got open sores and maggots are, like, growing in his arm, but he's, like, surviving. Like, somehow his body is creating some symbiote. Now it's, you know, he has a fever, or your body's fighting the infection.
It's doing everything it can.
It's doing everything it can, but your body always finds a way. Or you have to eat less, and then your body will find a way. You have to eat more, your body will find a way to balance itself.
It's amazing, isn't it?
Yeah.
It's amazing. So let's think about this, because you mentioned it earlier, right? About treating these complex lifestyle diseases with a pill, which is the standard approach right now, right? And that's medicine's built on that. And don't get me wrong, it can be very effective, right? Some of these things work. But sometimes it doesn't work forever, and it doesn't work for everyone, right? And so clinicians are starting to think about, are there alternative approaches, right? And is it possible we could use a dietary strategy? Now, obviously, we know dietary changes can just in and of themselves can help, right? So people with high blood pressure, for example, if you change your diet, you can actually start to reduce your blood pressure if you switch your diet appropriately. So we sort of understand that, but can we actually think about that and how that would impinge on the microbiome? Because this is what fascinates me right now and a lot of people in the microbiome field. And one of the reasons the Nebraska Food for Health Center evolved was to study this very thing, right? And to get at that question of how do we do that? So let me just give you an example of a study that's been done where we went from an in vitro system and went into one of these mouse models. And it has to do with starch composition. So now we're going to talk a little bit about the food itself. So starch, you know, is found in all grains. It's a major component of the grain. And starch is made up of two different types of complex carbohydrates. And one is known as amylose, and the other one's known as amylopectin. Okay? Amylopectin it's a highly branched, it's a polymer of glucose, so it's a bunch of glucose units that have been strung together by the plant. And it's highly branched, and it's highly digestible. Your body digests it very efficiently. Your enzymes can get on the outside, all the branches, and chew it up, and your body loves it. Amylose has no branches in it. It's just straight chain, right? And it's very stable, and it's fairly resistant to digestion. And so when you eat starch, most of the amylose is degraded by your body between the stomach and the small intestine. And even in the mouth, it starts breaking it down. There's amylases there as well that break it down. The amylose goes through most of it and reaches the colon. And in the colon, there are beneficial microbes, right? Microbes that you want to have there that love amylose. We call them amylolytic. I mean, they love this amylose.
They're like,
they love it.
It almost sounds like an alcoholic or something.
Oh,
It's
I
like,
know.
like,
Yeah.
give me,
But that's why I
give me
threw
my
it out
ammo.
there, these funny terms, right, that we use. So anyway, so that's starch, right? And the ratio of amylose to amylopectin is, you know, it's not really 50-50. It's probably more like 60-40-ish, depending on the...
For the pectin or for... What's 60-40, I guess? Does that matter?
The amylose to amylopectin, yeah, it's going to matter in a minute.
Okay.
So there was a mutant line of maize discovered back in the 1930s, and it's called waxy. And waxy maize produces starch that has virtually no amylose in it. It's all amylopectin. And this is a naturally occurring mutation. And in fact, it's been found in almost every grain species, right? So this isn't GMOs, nothing like this. This is a naturally occurring mutation that people found. Well, the food industry figured out that waxy starches have very different properties to them in terms of making foods. So their gelling properties are fantastic. So they make beautiful gels. And so the food industry uses waxy starches, introduces those into certain types of foods where this gelling behavior can really make a difference, right? So we did a study. We took 2 lines, and we used sorghum because that's what we happened to have on campus that one of the breeders was developing. 2 lines of sorghum that are otherwise genetically identical with the exception of that amylose content, right? So a waxy line and its parent, okay? And initially we took human microbiomes and studied them in vitro. So we just grind up the seed, we make it look like it's been digested, and then we feed it to that microbiome in vitro. And whopping difference between what the microbiome looks like with the waxy versus what it looks like with a parent that has good amylose in it, right? Very different. Now we ask the question, okay, that's in the test tube. What happens if I put those into a mouse now? So now we put those microbiomes, same microbiome, so same human subjects, same microbiome. We're studying in the test tube. We now put those into germ-free mice. And now we feed the mice a diet with either the parental line or the waxy line. And guess what?
Can I take a guess?
Yeah.
Without amylose, they got super fat. Or sorry, yeah, without amylose, they got super fat.
They're not super fat, but they got fat. They gained more weight. Yes.
For sure.
They gained more weight. And their microbiome was not the same. The microbiome looked different than the ones that were eating the...
Was it like less because your body was taking it all and storing it itself, correct? Is that something, almost a way to think of it?
It's a way
it's
to
like,
think
we got this is good to go.
So you had reduced levels of these organisms that you want, right? And because you're not feeding them, right? You're not feeding them what you would, one of the things that they like to feed. And so instead, all the starch is not digestible in the upper GI tract. Your body digests it and absorbs that all up, and you don't feed the colonic microbiome. Now, that probably doesn't matter to us humans unless you're eating snack pack pudding every day, right? Or something like that. But here's where it does get eye-opening. Yeah, there might be people that snack pack pudding is good stuff, don't get me wrong. But
there are a lot of things that you think that people don't eat that they eat. Yeah. I mean, we all do it, but there are things that you can't imagine.
But here's a connection, though, where it starts, it's an eye-opener. Because, so one of the grains where you would recognize waxy grain is with rice, because waxy rice is sticky rice. If you've ever had sticky rice, it's pretty yummy.
Ah, it's so good.
Yeah, sticky rice.
So good.
Now, there's only been a small number of these studies that have been done, but the ones that have been done are eye-openers, because if you go to countries in Asia where they're growing the rice locally and consuming it locally, right? Not growing and distributing like we do here, but they're growing it and consuming it locally. There's studies of where they've gone in and they've looked at those rice cultivars that people are actually growing like that, and about half of them are waxy. They have the waxy characteristics.
So this is going to give you a perfect, this is like the perfect thing to look at.
Yeah,
perfect data point.
So think about it. These people are eating it every day. Eating it every day. So
yes, a lot
you start to scratch your head and say, boy, I wonder if there isn't a health outcome. Right? But again, this starts to teach us, okay, how can we modulate the microbiome? Well, so at the Food for Health Center, we start thinking about, okay, how can I use that information? So now we're going the other direction. So, okay, waxy isn't good for you, but there's also another mutant in maize that was found a long time ago called amylose extender. And it makes lots of amylose. So most of its starch is actually amylose.
So instead of like 60-40, it would be like an 80-20 or something.
It's almost 90-10.
Okay.
In some, depending on the cultivar, right?
I feel like
depending
it's
on the
sodium
line.
husk.
And again, naturally occurring mutations.
Yeah.
And again, the food industry has capitalized on this also because the starch that comes from this high amylose starch, oh my gosh, it doesn't absorb water at all. I mean, you can't form a gel with it to save your life, right? It has a very different behavior to it. And this it's called high maize is one of the names of one of the products that's out there, and can be used as an ingredient in things.
What would it be used for?
You might use it in cakes and breads and things
to really dry it out.
Yeah, where
gelling's not good.
Yeah. So now we've got the Food for Health Center spent the last 3 years growing these 3 lines of maize, 3 lines of corn that again are otherwise genetically identical. There's a parent, There's a derivative of that parent that's waxy, and then a derivative of that parent that is the high amylose, right? Amylose extender. Otherwise, they're genetically identical. Only difference in those is the amount of amylose in the starch.
Okay.
Right? And I should have brought some of the seeds with me from that, because we've now...
Do they look different?
No, they look. You couldn't tell. As I said, it would have been great to bring the seeds and show you. Right?
Oh, okay.
We just had a visit from one of our donors, and we went through a whole demo with us to show them how differently they behave. Right, but to look at them, you can't tell the difference. So we're asking the question now: can we use that high amylose stuff to have a positive impact on the microbiome? And we're headed into a human clinical study with that. That's in the works right now. It's just starting to. We're getting everything ready to get that rolling. But it just gives you an example of how we take that information from what we learned from an experimental system, and how can we move that now into a food and a food situation where we can actually try and manipulate the microbiome. And in this case, we're using a whole grain. We're doing it from a whole grain. We're not purifying, extracting it, anything. It's the whole grain.
Did you literally just put, explain that? You take the whole grain and
just grind it up into
grind
a
it
flour,
up, and then is it just inject? How are you getting it into the
so
mice?
That was a bit of a challenge too, because
it feels like it'd be tough to get in.
The properties are so different.
Yeah.
Right? So
the one they would have loved for sure. Waxy, they would have been like,
oh, that one.
Let's
was
go.
Great.
Give me some more of that.
But this high amylose stuff. No, we incorporated it into a biscotti.
They would have crushed that.
Yeah,
I would crush it.
and
you don't want to give it. I would have been a great trial subject.
Yeah. Well, I'll let you know when that study starts, right? Because they actually don't taste bad at all.
Yeah.
You can kind of tell the difference because the
I feel like the
high amylose
90-10 one you would. Yeah,
it's a little more gritty, but otherwise and you could easily eat 3 of them a day. And in doing that, you get roughly a 20-gram dose, of amylose from the high amylose. And so there'll be a really neat study that's going to start with those here soon. But that's just one example. Another example of what we've learned, and again, here we're using an actual mapping study where I don't know what's affecting the microbiome, but I'm using plant genetics to figure it out. And so we...
Okay, so explain that. So you have plant, you know what's in the plant. That's the idea.
Genetically, I know.
Genetically,
right?
you know what the plant is.
And I know there's variation, and I can find the variation, but I screen hundreds of those lines, right, that are genetically different from one another. And then I identify lines that affect the microbiome using this in vitro system that we use. And then I can go in and genetically say, okay, what is it in that region where the variation is in the plant genome that's affecting the microbiome? And by doing that, we landed on condensed tannins. And again, this was in the sorghum population that we were studying.
Condensed tannins?
Condensed tannins, yeah. So
I only think of tannins when I think of red wine.
Yeah, and that's what everybody thinks of, right? So tannins is a whole class of molecules. It's not just one type of molecule. They all have some similar structure to them, but condensed tannins have a very specific structure to them. They're very large, complex, what we call polyphenols.
I didn't know this because Baobab has a ton of polyphenols, which are tannins, but it's dry powder, so it always freaks me out thinking about tannins come in different forms.
Yeah, they do. They come in very different forms.
Yes.
So sorghum and millet are one of the few grains that actually make tannins, and we believe it was an ancestral trait that was actually bred out of the grains because it gives them a bitter flavor. But it has been maintained, I'll tell you.
The only reason I ask is I feel like sorghum and millet would have some sort of brewed substance, because it seems like tannins from a brewing to distillate or brewing process help it because they're brewing out the bitterness.
Yeah.
With the yeast, this doesn't make any sense. Well, it does make sense, but I don't know how this works. So somehow having a yeast with this turning the sugar into alcohol would somehow take out the tannin bitterness or something.
It can reduce it. Let's say, right? It can reduce the bitterness, right? But anyway, sorghum maintained this trait, and it makes what's called condensed tannins, and using this mapping strategy, we found that those condensed tannins have a whopping effect on the microbiome. Oh my gosh, it's like one of the strongest effects I've seen on a microbiome.
What do you mean by effect?
It stimulates growth of some very beneficial microbes, right? And this fascinated me because
Was it the specific? Do you? I guess you're getting there, but do you think it was the specific tannin?
yeah, we went all the way to showing. We went all the way in that paper. We went all the way from doing the microbiome screening, identifying the genes in the plant, identifying the function of those genes, and then showing that we could actually take purified condensed tannins and throw them into that microbiome and get the same effect that we were getting with the plant. It was fascinating. And so there's a certain microbe called Faecalibacterium, and so we're fascinated with this. And the reason this one gets our attention, so Faecalibacterium poisonitii is its whole name, and we just call it FP.
One more time.
Yeah, Faecalibacterium poisonitii. It's just fun to say.
That sounded like a mixture of Italian and German at the
German,
end.
yeah.
German wanted to get in there.
We just call it FP, right?
Yeah, I like FP.
FP's a lot better. Yeah. So FP is considered a beneficial organism. It's one of these it loves amylose also, right? But it's one of these organisms that produces from complex carbohydrates such as amylose. It produces a short chain fatty acid metabolite, and that metabolite is called butyrate. And butyrate is considered a very beneficial metabolite because it does a lot of things in your GI tract, especially in the colon. But one of the things it does is it actually feeds those colonocytes, those cells that are lining. They make that very thin layer, right, separating.
Does it make the layer thicker?
No,
it just feeds them.
It feeds.
Keeps it healthy.
They use it for energy. The human cells use that butyrate as an energy source, and there's a few other cells in the body that will do that too, but those cells are adapted to doing that, right? And so it's this mutualism, right, between the microbiome and the epithelial cells lining the colon. So, butyrate producing beneficial organism. We also know that this organism is depleted in patients with inflammatory bowel disease. So it's gone. It's hard to, their microbiomes are missing it, right?
Like every single one.
I don't know about every single,
but like literally every time you have
this.
It's
a signature, yeah. If you got somebody with IBD, the chances are they're missing this organism. They're missing others too, right? But they're missing this one.
Okay.
And we also know this is the field, not our group, but we also know that this organism can stimulate and tickle the immune system as well in ways that we don't yet quite understand.
Never heard that term, by the way. Tickle the immune system.
Yeah,
I like that.
A little tickle, right?
Improve it.
In a good way.
Yeah,
right?
like
Yeah,
get it going.
Yeah,
get it going.
It makes it do things you want that are anti-inflammatory.
Yeah.
Right.
Okay.
So, think about this now. Here you've got an organism that is associated with reduced susceptibility to inflammatory bowel disease. Mm-hmm. Now all of a sudden I've just found something, condensed tannins, a way to selectively stimulate that. And there's other organisms we see as well, but that one in particular really responded to it. Now I've got a way to selectively stimulate that organism. Yeah, that makes you think about how I'm going to start breeding to produce even higher levels of tannin. Oh, and maybe I talk to the breeders and say, you know what? If I could have sorghum that had high levels of tannin and had high amylose, now I've got two things that I can use to start modulating that. And I'm just doing it by breeding and doing it with a whole grain strategy. So fascinating. So you can see how the mind starts going and thinking about how you can use these dietary strategies to modulate the microbiome.
But I think you're one of the few people that is really focused on, and I guess that's not just in the sense of nutrient density, where people a lot of times think of food as a nutrient-dense fruit, not nutrient-dense food. It's like you're saying this food can produce specifically important benefits to not just say nutrient absorption. It's the opposite. It is nutrient absorption, but it's providing specific nutrients for the bacteria within the system.
Yeah. You're feeding the whole ecosystem, right? And that's kind of the idea.
So do these exist yet? Do these types of products exist yet?
Well, we're just in the process of starting to do these things, but it just gives you an idea. This is sort of a roadmap for how the Food for Health Center goes from discovery to translating these into things that are important, right, for our health. We're one of the few places in the world that reaches all the way back into plant genetics. a lot of people, remember I told you early on, a lot of people are using purified fibers, right?
Explain
or
the difference.
Fibers they've extracted. Well, they're just extracting, fiber contents out of the plants. And I'm not saying that's a bad approach, right?
Is it probably easier? Yes.
It's easier in a way, but the
conclusions can't be as good, but it's easier.
But the downside to it though is, so let's say I take that, and Baobab's not a great example, but let's say I take.
Well, say like a psyllium husk.
Yeah.
What was another one that's like the inulin's been used a lot right now?
Inulin's been used a lot, right? So, and it has beneficial properties. Don't get me wrong. As a prebiotic, it functions well. But these are extracted and enriched out of whether it's chicory root or Jerusalem artichokes, or there's different things where you can get it.
So now they take that extracted fiber, because that's really all they're using from those products anyways.
Right.
And then that's now become inulin.
Right.
And then they're studying that specifically.
Yeah.
Okay.
The effect of that on the microbiome and or health characteristics, right?
Okay.
And inulin is one of the best studied, and we know it clearly.
Yeah, I mean everybody's using it as a prebiotic because of it.
Clearly has prebiotic benefits to it.
Yeah.
There's no doubt about it. But the one thing about that approach is that your microbiome is not seeing it necessarily in its natural context.
Well, this is where I did want to bring up Baobab. So I will say this. I'm a big proponent of whole food. So when you talk about changing characteristics within a whole food or a whole grain, that fires me up more than saying, let's extract this little bit of this and then this little bit of this. I'm like, how do we make products or find products that exist and make them better as the whole food?
And that's exactly what we're trying to do. That's why we reach back into plant genetics and say, can we manipulate that through traditional breeding strategies to improve these traits that we know in the whole food will have an impact on the microbiome, as opposed to this extraction approach. And don't get me wrong, some of these extraction strategies and purified or refined ingredients have clear benefits.
You're saying you got I'll use refined. It's refined. And now you have this inulin, and then your body, it's not the thing that it's used to. It's like, what is this, right?
Yeah.
Or is it not? What is it? It can use it, but it's because it obviously has benefits.
But and this we don't know yet. We don't know the answer to this yet. We're going to learn the answer to this in the next few years. Does the body see it differently? Does the microbiome see it differently when it's in its whole grain context versus, put into a drink, right? Does it see it differently? And I don't know the answer to that question is, but I suspect it does. I suspect it will see it differently.
Well, because I do want to bring this up. Not yet. Well, I still want to go on this. So they're studying it in the sense of they're going to do the same studies in vitro, and then they'll do it in a mouse as well.
Oh, that's so you can go into that mouse model if you want to, or some of these things you can go straight into humans, because you're talking about food, right? You're talking about food, it's natural.
So the trials are different.
Yeah. I want to go back to the tannins, so I wanted to tell you one more story about that. It's kind of like a waxy story, right? So, going back to the tannins, remember these condensed tannins in sorghum stimulate very beneficial organism, right? Faecalibacterium, FP.
FP.
FP. You got it. Yeah, you got it. FP.
There's a lot of words I don't know. That's why I'm talking to you and you're telling me. Yeah.
FP is a good thing. HMOs are a good thing, right?
Yeah.
But so in Africa, which is a primary place where sorghum is consumed almost on a daily basis, right? There's this fascinating phenomenon, again, of where crops are being grown locally and consumed locally, okay? In Africa, there's a bird species over there called quelea, and quelea loves sorghum, but it does not like high tannin sorghum because it's bitter. And so, the density of quelea varies by region in Africa. And so in areas where the bird population, this quelea population is really high, they're growing high tannin sorghum. And people are eating this every day. And then in areas where the bird population is low, they're growing the lower tannin sorghum that tastes better, right? And they're eating that every day. Again, I just wonder, is there any sort of disparity in health outcomes?
I totally want to know.
I don't know.
Where is
figure out
that?
how to study that.
Is that just all over Africa?
It's all sub-Saharan. It's in the sub-Saharan area. Yeah, and I think it's if I remember right, the qualia are higher in the eastern and lower in the western and southern portions of that area. And it gets even more fascinating. So there's this fantastic group that studied this in Kansas State and in China, and I think in Washington State as well. Big collaboration that studied this. Really fantastic paper. The story gets even better because they also scratch their head and ask, well, how are the humans consuming this bitter flavored stuff?
Mm-hmm.
Turns out that if you study the taste receptor genetics of the humans, right?
Mm-hmm.
The humans that live in the area where they're growing the high tannin sorghum have a certain variant of the bitter taste receptor where they don't taste the bitter. Versus the people that are growing.
They develop it, or is it?
Yeah,
they don't know.
And that's the big
on?
question: is this a migration thing? Is this an adaptation thing? We don't know the answer to that.
Don't study Americans on that one.
Oh no, but
We could do
but
the
think
sweet
about
receptor.
it. Well, but think about it. I had my 3-shot Americano. I like my coffee bold and intense.
You
coffee
develop
flavor.
the bitter blocker.
Well, I also like. Guess what else I like?
Sweets.
Really, no, not like
I don't know.
really sweets. But no, another thing you can, if you like really high intensity coffee like that, you're probably gonna like in your face bold red wines too, which I do. Again, high in tannins, right?
Yeah.
And chocolate preference.
Black,
Love dark chocolate.
Yeah.
So you can see how those taste receptors influence, right? What you
those
might
for
like
sure
or
can be
not.
developed. Although it's interesting because I would want to know in the younger population how they struggle through it. Because I feel like no matter what, until you're like 14, you're like, please don't make this bitter.
Oh yeah.
Anything sweet would be better. But I think
I don't know enough about that to know.
no. But if you grow up
but
I mean, these are all. This is what I can talk because it's just it's just completely anecdotal. But it's like it seems like before they have to make it a little bit different for them, and then they eventually can consume it. But I think if you grow up with a specific taste, you don't ever have a problem. You probably develop that right away.
Yeah.
I think of that because that reminds me of, oh man, I don't know when he wrote that book, The China Study. And I think about that first case study of the idea of like vegetarianism and then looking at places that consumed less protein in Western China versus Eastern China. And then from there, it was like, okay, was it plant protein, meat protein?
Yeah, right.
When you start to have these populations that you can really see the differentiation. And I just, the tannin thing's really interesting because it seems like, as tannin usually is grown as well, it seems like most high tannin products or high tannin foods are grown in really harsh climates.
Yeah, because the tannins actually they help the plant adapt to stress. So that one of the reasons the plant makes them is it helps stress adaptation. So when the plants are put under drought stress, for example, their tannin content will go up because it helps them deal with that. Now, it's not clear exactly how it helps, but it definitely helps.
Yeah, because I heard somebody talk about that with olives. So olives have that. Baobab grows in the most semi-arid places.
Yeah.
And I think it has it has one of the highest polyphenol. But in the case of tannin versus polyphenol, that's not...
Tannin's a type of polyphenol.
It's a type of polyphenol.
Polyphenols are a whole category of molecules, and tannins are just one type, and then among the tannins, there's different types of tannins, right? So it's very complex, but they all share a similar type of structure, a similar type of chemical structure. There's some similarities.
Have you found that most tannins do something in a positive effect, or some are different?
Well, it depends. So, the funny thing
maybe I'm hoping this for red wine.
Well, no, the funny. I know.
The tannin, and it's like the Restrovial deal
That's
with
the
red wine.
resveratrol. Yeah, you want that. That's a good one. No, tannins generally is funny. Tannins generally have a negative connotation or negative context in terms of animal feed. Oh, this is another fascinating thing. I should have told you about that.
But that seems to make sense now because in animal feed, you want the animal to get as big as possible, as fast as possible. So I want the pectin, amylase.
No. It's the tannins in there. So what tannins do? One of the things tannins do, they bind everything. Oh my God, they bind everything. You can take tannin and put it into a protein mix, and it'll precipitate all the protein out of there right away. It's amazing how these tannins do this. So again, I come back to the natural context, right?
Yeah.
That the organisms see the tannins in. So tannins are thought of as anti-nutrient.
That's what I. Yeah, that's what I'm.
That's
right.
that's the way
Like,
they're thought of in especially in the animal production industry. If you're gonna put sorghum into animal feed, you're using low tannin sorghum, right? Because the animals won't gain as much weight if it's high tannin. Now, think about it. That's not a bad thing in the human population, but from animal production, it is a bad thing, right?
but would the animal be a more robust animal, but
and
smaller?
That's a good question, because here's the thing. Here's the thing that fascinated me, right? When I looked in the literature and found this, it blew me away, right? So we saw this certain signature in the microbiome of tannins affecting a couple of other organisms, right? You see that same signature in animal microbiomes, including swine, poultry, same signature, same group of organisms, all right? And so you start to scratch your head and you start to think about, all right, maybe some of these gut organisms have literally evolved, and that's their niche, is to be able to break something down like a condensed tannin. And maybe it's not even the condensed tannin that they want. It's they want whatever the condensed tannin is actually bound to.
Huh.
And if they can break it apart, they have a selective advantage to being able to get it. It's almost like a little nut, right? This tannin and this protein complex is like a little nut coming in, and only certain microbes can break the nut apart and get at the nougat inside. And if you can do that, that gives you a selective advantage to growing in that ecosystem. So it's fascinating to think that, and we don't know if that's true or not.
Yeah,
We're starting to study that now, but it's fascinating to think about that as, wow, that these things might have evolved to actually be able to do this, and there's a benefit to doing that. But the practical aspect of that is that we can use plants with enriched condensed tannins as a way to stimulate very beneficial microbes. We can use plants and whole grains that have higher levels of amylose in them to, again, enrich for beneficial microbes.
Does the only thing that I think about is right now everything's explained very simple and like simplistic terms, or it's broad terms in the health space. So you have gut health, microbiome. You might say sugar, like glucose, fiber, right? But the way that you describe it is this ecosystem, and there's a larger benefit to the whole food, or what the potentially specific ingredients within that whole food can do to your entire system. It's almost like you can't, it's not quantitative, or like in how it can actually benefit you. And so describing it to a consumer seems very difficult.
It's a challenge, right? That is a challenge. You've started to wrap your head around this because you have a real interest in this area, right?
Yeah.
But your average layperson out there, it's a challenge to explain to them why this would be a benefit to them. But I think the cool thing is I need to introduce you, and hopefully Peter will forgive me for this, but I need to introduce you to our colleague, Dr. Peter Mannon at the Medical Center. So Peter is a member of our Food for Health Center. He is a gastroenterologist and a well-known researcher, world-renowned researcher, clinician, who largely looks at immune dysregulation in inflammatory bowel disease patients. But the other aspect of Peter, and the reason why he's a member of the Food for Health Center, he is very interested in, can we actually use dietary strategies to keep patients in remission, right? Not necessarily people with acute full-blown disease, but people in remission, can we keep them in remission longer simply by a dietary strategy? Because that's never really been tried. And so you're starting to see the community, the medical community is starting to wrap their head around this. The funding agencies, so National Institutes of Health, for example, has Nutrition for Precision Health initiative, right? And so they're starting to think about this as an approach. Now, they're not necessarily going all the way back into the plants the way we are, But Peter's the one that's going to, remember that study I told you with the cookies? He's the one that's going to lead that first study, right? And so you're starting to see this transition where people are starting to scratch their heads and say, well, maybe there's something there. If we can figure out how to manipulate that microbiome, and we're still learning, right? But
Yeah.
the key for my approach and the center's approach right now is to let's fill the cabinets, so to speak, our cabinets of tools, with foods that we know can perturb the microbiome in a very specific and beneficial way. And then figure out how can we use those foods and introduce those into diets of patients, and can we start to understand based on their baseline microbiome configuration who's going to respond and who isn't going to respond. Can we start to predict that? That's the fascinating path the field is going down right now. And yes, there's still people studying mechanisms and all that stuff, but if you want to look at this diet microbiome modulation area, that's where the field's going, and it's an exciting time to be in the field right now to see which way this all goes.
Yeah, and then so does that mean you have to work with food producers? I mean, you're kind of tied to everybody in the sense of trying to work with everybody.
And this is the benefit of something like the Nebraska Food for Health Center is our mission is to unite agricultural medicine and biomedicine, right? And we do that through the gut microbiome. You see, it makes sense, right?
Yeah, it totally makes sense.
Yeah, but that's our mission. And so if you look at, we have what we call our research platform. It goes all the way from this, molecular genetic discovery that we do in the plants into these translational animal models where we can test an animal model, and then all the way into, okay, can we scale this up and get it into a food? It's easy to put it in mouse chow, but can we get it into a food that a human would eat? And now let's do clinical studies in humans. We span that whole gamut, and it's not unlike the way drug companies work, pharmaceutical companies work. Going from in vitro discovery to maybe an preclinical animal model, then into the expensive clinical trials. But that's our overall approach to doing that. And it takes people with all different types of training. So if you look at our center members, we have plant geneticists, we have microbiologists like myself, we have food chemists, we have immunologists, we have food scientists, right, that are actually help make food. And then we've got gastroenterologists, and we have clinicians, we have nutritionists, right? All of these people working together. None of these projects that we do are driven by just one investigator. It's a team, right? Somebody's usually the lead on it, right? But it's a team of people that are
based on whatever section you guys are focused on at that time.
Yeah. But it's a team of people that are contributing together. Yeah, it's really cool to see that.
And then what do you think, generally I understand the mission now is to connect them, and then this is to, I guess, what we talked about a little bit, what spurred your desire to do it this way? Like, do you want to just see us consume food different? Do you want just healthier populations? This is also just the most fascinating subject probably possible because of the things.
Well, it's fascinating, but the way that center evolved, right, was through a process. But remember, I told you we were a gut function initiative of 4 guys having a lot of fun together doing science back in the early days, right?
Yeah.
And now it's 30 members, and if you count the students and trainees, it's 60, 80 people.
Wow.
It gets pretty big, right? But what drove us to do that was largely, to keep it in short form, was largely we had a philanthropic donor come to the university and challenge us and the university, and the chancellor at the time was already talking to us and was already challenging us to sort of morph into something more than bunch of people having fun together doing research. Right?
The university system? No.
Yeah. But they resourced us, right? And so they challenged us to create this vision, right? And the philanthropist was Jeff Rakes, and he literally said, create agriculture 2.0. and that's where that vision came from. We were already thinking about, because we knew diet had a big impact on the microbiome, I was already thinking about, is there a way to get back into the plant genetics? So we have this powerful plant science innovation center here on campus. How do we take advantage of that? How do we put these pieces together? But it was that philanthropist that put the challenge and put the money on the table. And then the university also put resources in on the table to make all this happen. So that was a motivation, but we were also motivated by doing something people haven't done before. Doing something new. Nobody's ever done this before.
The
right?
2.0 makes sense. So then because there's a lot of talk now, right, about regenerative farming.
And that's all important. Don't get me wrong.
But this isn't that, right? Like that's super important. But it's really interesting because this is another layer of the monocrop, but with specifically not. I can't use the word nutrient dense. Right? It's not nutrient. It's more than that.
But if you think about what factors have driven plant breeding and improvement programs. Production, yield, sustainability, right? Sustainability is the new one on the block, right? Nowhere in there is nutrient content. Health has not been a part of that picture at all.
Well, and this is so.
With the exception of maybe some global, right?
But not okay.
It's not been a big driver.
I think for us as a civilization, though, it's been forever. It was always like, how do we produce enough? We're in the first time in the history of the world where you're like, all right, we produced enough food.
Yeah.
Now we need to make sure that what we're producing is actually beneficial
Correct.
for us, because
Correct.
forever it was just like, we need to produce enough food, so that would make sense. Whatever's going to absorb the quickest is going to be glucose for energy to work for 12 hours a day outside made sense. Now we're at this new stage where it's like, okay, what produces the most, energetic human with the least amount of resources, but also, like I said, it's not nutrient dense. It's more than nutrient dense.
It's
like,
the wrong nutrients. That's the problem.
Yeah.
So one of the things that's happened, not necessarily with breeding per se in crop improvement, but in our food supply, is our fiber content is abysmal. The average individual is eating 12, 15 grams of fiber a day when the RDA is 28 to 30.
I just talked to David Newton a couple weeks ago, who's a gastroenterologist in town, and he was like maybe 5 to 10 for a lot of people. Maybe 5 to 10.
Yeah.
And like our product has 3 grams per serving, and it's really interesting because in most drinks you have 0 fiber, right?
Yep.
But it's really interesting to hear people even think about having fiber in a drink or fiber in
Yeah.
And it's because and I don't know, and maybe you could speak to this, but fiber got a bad rap at some point as being something that doesn't it only got associated with going to the bathroom since the 30s or something. I don't understand.
Yeah, and I don't know when that happened either. I know what you're talking about, right? It's like, fiber is something you drink when you're old people, right?
Yeah.
Metamucil, right? It's the whole Metamucil story.
And I feel like they probably started that in the 30s because that's when I think they lost.
But the problem is that fiber is what feeds the microbiome. The microbiome they thrive on fiber. They love it. And if you cut that out of your diet, now you're switching over to organisms that instead are growing on protein and fats, right? That's not the same organisms that thrive on fibers.
Two things on this. So explain fiber for... No, don't explain fiber. Maybe explain fiber.
We need to a little bit here.
Explain this because then I also want to get into... Because there's people now that are doing like the carnivore diets, meat diets only, or paleo, where there's very low fiber. And that's really interesting what happens in the microbiome. But some people it seems to have benefited them. So anyway, start with fiber.
Yeah. So fiber is not one thing, right? It's like the polyphenols, even more complex, right? Fibers are very complex, and so you can kind of separate them into what we call cellulose and hemicellulose. And in plants, they form part of the cell wall. They're the cell wall structure in the plants. And cellulose, for the most part, our body doesn't break down, nor does our microbiome. It's not evolved to do that. But our microbiome loves hemicellulose and it loves starch, right, which is another fiber component that's not a cell wall component. The hemicelluloses are extraordinarily diverse. I could give you all these names and blow you away, right?
Can you give me a couple? Because I think
Arabino's island.
Okay, that makes sense. So Baobab has a ton of them.
Yes, it's got a lot of them. Baobab has a lot of these things, a lot of these hemicellulose components in there.
Is that because of being do fruits generally have more?
Well, fruits also have pectins too. which you don't
Right.
find in grains.
You won't find that in a grain.
You know, you won't find. Well, you'll take that back.
Some.
You'll find some, but not nearly the amount that you'll
Okay.
find in fruits, right?
Okay.
So the fiber structures are different. Is my point.
Yep.
There's a tremendous diversity in the fiber structures, and depending on which organisms you have in your microbiome, and a lot of that's going to depend on what you eat a lot of. You'll have organisms that can degrade. So let's say you eat a ton of fruit. You'll have organisms that are good at degrading pectin, right? Because there's a lot of pectin in there. And glucosylans and or xyloglucans and other components that are enriched in fruits and fruit cell walls, the fruit fibers. And if you're eating a lot of grain or if you're eating a lot of greens, let's say, it'll be a different category of fibers, right? But in general, it's these fiber components, hemicelluloses, that make it through the GI tract, and they get hydrolyzed by the acid a little bit. It breaks, and they're very complex structures, right?
Okay.
And so the stomach acid will break them down a little bit, but not really in a way where you can extract any energy out of it or any of the nutrients out of it. They make it down into the colon, and there the microbes are living on it. And those microbes, the beneficial microbes, the microbes that are we think are the best for us, they love those things. They love those components. And the problem is we don't eat that stuff.
As the term was said,
We did a few thousand years ago.
Yeah, it's not in processed food.
Yeah, a few thousand years ago, that's all we were eating. Think about that. We were eating a lot. You know, the hunter-gatherer diets. We're eating a lot of that kind of fiber.
Yeah, the Hadza tribe's been studied a bunch.
Yeah.
I think you probably know them, but
Yep.
with the extremely diverse microbiome.
Yes. So it's very diverse. There's organisms in that microbiome that you do not see in ours. Right? They're not present.
If you were to go into their, like say the Hadza tribe, and I started eating what they ate, would I all of a sudden develop that microbiome?
So that very study, so my good friend Jens Walter was just here. Jens is one of our colleagues. He used to be here at Nebraska many years ago. He just did a study very much like that. And it's not published yet, but he presented it when he was here a few weeks ago, and it was fantastic to look at it. They actually created diets that looked very similar as best they could to what the tribes were eating. And one of the challenges in that is the increase in that type of fiber in you and me.
Oh, gastral discomfort.
Has an effect. Yeah. We were joking about this the other day with Dr. Dehan, who's one of Jens's proteges, right? Who's on our faculty now. You know, what a great party trick, you know, to show up with like, hey, I got this great source of fiber. Let's all try it. You know, and within half an hour, everybody.
Yeah, it's struggling. Well, because with David, we were talking about that. And I said, "Is it incremental?" He's like, "Yeah, you have to literally.
You got to walk yourself.
You got to get yourself there." Because I said, "There's a fine line between like benefiting your gut and bloating."
Yes,
there is. Or like satiation and bloating.
Yeah.
And it seems like that's where people really struggle. Is that you go from zero to the maximum. And he said the best way, like. Because he prescribed, he likes psyllium husk a lot for patients that have no fiber and that having not digestion, well maybe digestion problems, but going to the bathroom problems. And he just usually tells them at night because hopefully they're asleep for a lot of the discomfort, I guess. But I thought it was really interesting. So yeah, that would totally make sense. So they go from none, because I know the Hadza tribe was like 75 grams of Baobab or 150. It's like 150 grams of Baobab per day, which is the equivalent of, it's 50% fiber, so 75 grams of Baobab just from Baobab. Then they were having tubers, and I don't know what else. And I think they even roots, like they would just have roots as well.
Yep.
Which is literally not digestible. That would just be a literal, that's not even insoluble. That's just like a fiber. That's like a root. That's like almost like an inulin style.
Yeah. Well, inulin can be digested by the gut microbiome.
See, that's what's so different. Yeah. So I guess there are
components.
Roots.
Yeah. There's components of the roots that can be, but again, a lot of that's going to go right through. But
yeah, which would help, I guess. But you don't.
But if you're going to switch to a high fiber diet, you better walk yourself up to it.
So is that was that one of the findings as well? Was like, okay,
Yeah,
when we do this
and
study, we're like, we need a month to
But
prepare.
they did, yeah, they did. So they planned it out fairly well, where they did walk people up on it, right, and so that they were able to tolerate it. But they said, it definitely is a challenge to just start eating that right away. Right?
But I imagine, would they have studied how they felt as well with a high fiber diet?
Oh yeah. So bring Dr. Dehan in, because he was part of that study.
Yeah,
He can tell
I
you all about it.
would love to, because I
He can tell
find
you about it.
I've consumed a lot more fiber now that once we started Tiiga and just consuming Baobab, because I think Baobab on its own and then Baobab and just Baobab in general has become one of my main fiber sources, and I feel completely different when I have it and when I don't. And there's this weird feeling that, because like I'm trying to think where else. Like I consume beans a lot for fiber.
Which is another good source.
Great one,
yeah.
but they don't make me feel. It feels different, and I think beans. I know that obviously there's the joke about the musical fruit, but beans have another substance in them, right? That creates a more gaseous, like the way that it's getting consumed by the microbes.
So they have these. They're called alpha galactosides, and so there's three main ones: Stachyose, Herbiose, and I forget the third one, but those are well recognized for their gasogenic properties. Yeah, the problem is the gut microbes produce gas out of them. That's the challenge with them. And you stimulate the organisms that have those and have the pathways for breaking those down, then you quickly stimulate gas production with those.
Got it.
So that's one of the downsides to beans, is that they do have fairly high amounts of those. But they have other things in them that are very beneficial as well.
So if you were to say other fibers that, because obviously like the sorghum hasn't come out, would be considered with the high tannin would be a high fiber product, correct?
It would be relatively high fiber. Not the tannins themselves; they're polyphenols, but
so they don't register, because like we don't right now have a way to. I mean, the ORAC counts as without count under the polyphenol count. Like there's no way to tell people that there's a lot of polyphenols, correct, in a product.
Well, you can tell them, but they know what it means.
They don't know what it means, and then there's no way to like register that on a scale, right? Like, you know how many calories are in something.
Well, you could measure it, but right now, and I've been talking to food producers who are looking at sorghum-based products, right?
Yeah.
And that's the question I know. How much do we need? And I don't know right now. I can't tell you what amount you need. Give me some time. Give us some time to figure that out. Now that we know that it's a player, then we can start to figure out, okay, how much of it would you need, and what proportion of the population would respond to that sort of thing.
For an efficacious amount per serving.
Yeah, so I don't know the answer to that.
I would love to get to the point where on a label or even the front of a packaging, instead of putting these basic nutritional facts, it's things like this, but like explain. And I don't know how you'd explain them simply, but it's a really interesting. It's going to be a very difficult challenge, because at the end of the day, people just want 300 milligrams caffeine, right? Like,
right.
Oh, I don't feel good. Let's just have more caffeine, or I'm tired. And it's like, oh, actually, if you had this tannin that this microbial could use, they'd be like, they'd be falling asleep.
Yeah.
But that's why I think having a conversation for a while about this, getting into them, and then being able to put clips together as well on the side and explain it in shorter form can start to make a little bit of sense. But I think these are not simple things. But I think people are thinking about their health differently now, right? Before it was like, oh, I don't feel good, go to the doctor, I'm done. Now it's kind of this combination, and again, you probably know this too, but it's like you have the biohacking, you have everybody in between, but in general, people are starting to pay attention to their overall health. So these things are going to be easier to articulate. I mean,
I'm hoping.
I just don't know how still.
Yeah, I mean, there's still going to be. There's still going to be a chunk of the population where it doesn't matter what we say.
Yeah.
Right?
Yeah.
And I think to some degree, it's on us to change how the food industry thinks about it, because they're the one that enables that chunk of the population, because they produce stuff that we don't necessarily should be eating every day.
Yeah.
Right?
I mean,
how do you change that? Good luck. But I think that's a key place, right? And I think the bigger challenge for people like yourselves, with companies like yours, is educating that consumer, right? What is it you can tell the consumer about your product that they will understand, and they'll understand why it could have a benefit to their health? Because you're not allowed to put a health claim on the label, are you?
No, and I don't want to put a health claim, but I've been trying to figure out how to describe, and this is just an example, and maybe you can help me with this. So the idea that most foods are pasteurized, which is not a bad thing. That's a good thing. It kills bacteria.
The bad ones.
The bad ones, because you study this, so you know this.
Yeah.
But the Baobab fruit's super unique because it's naturally powdered, and it's able to not have a secondary kill step, and then you're able to consume it directly. And so I'm trying to describe the benefits of consuming something that's high in polyphenols, high in fiber, in its natural form.
Yeah,
and
I was talking about earlier. Yeah.
And I don't know how there's no like 3-word slogan to describe that, right? But there kind of is, because it's not, the raw is not a right word, because raw has a weird connotation sometimes for people. And nutrient dense, you've been making me think about nutrient dense all day today, because nutrient dense doesn't sum up the idea.
Not really.
That
it's
like bioavailability seems like a way better word, but it's still too long of a word.
That's a scary word out there, probably,
yes.
too. I mean, it's, nutrients in their natural context, right? Yeah, nutrients in their natural context. That's really what you're talking about, and
that's why I love Whole Foods. Like, I just feel like Whole Foods.
Yeah.
Like, anytime you eat a whole food, you can tell the difference between anything processed.
This is what we grew up as a species eating.
Yeah.
Right?
Oh, I know we're running out of time, so I'm going to let you get out of here. Thank you for coming, Andy. This was absolutely amazing. I got so much information on this. Hopefully people that are listening will get the same, and we will see you next time on the Baobab Project.